All right. So when I say polyacrylamide gel, so electrophoresis in polyacrylamide gel is commonly called as what? PAGE, PAGE, right? So this is the abbreviation, polyacrylamide gel electrophoresis. So in this, you are going to have the support matrix or the gel, which is formed from polymerization of two components, that is acrylamide monomer and smaller amount of NN methyl methylene rather I should say bisacrylamide. So bisacrylamide and acrylamide are the two major components, and bisacrylamide is essentially two acrylamide molecules. So if I have to say ki what is bisacrylamide and what is acrylamide, so acrylamide, methylene, acrylamide will form bisacrylamide. Bis is to two, right? So two acrylamide molecules which are linked to methylene, they are called as bisacrylamide. So acrylamide monomer is going to be polymerized, and how the polymerization is going to occur, that we will see in the next slide, right? So that polymerized acrylamide is going to form the gel, and the pore size of the gel is going to be dependent upon the quantity of gelling material that you add while preparing and casting the gel, right? So higher concentrations and increased amount of bisacrylamide will give smaller pores, that we know, and vice versa also is true. So depending upon the size of the molecule that you want to separate, you have to decide the quantity of polyacrylamide to be put in or suspended in the buffer. Then it is brought to liquid uh, um, conditions, and it is poured at so on cooling it is going to gel. All right. So there are two main types of the gel, two parts of the gel that you will see when we talk about polyacrylamide. I'll get back to that. But before that, let's see the next slide, what I was talking about, the structure. I know this slide is a little, what we say, uh, 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 dark to see, but of course the structures are very well seen here. You can very well see CH2, CH, CH2, CH, CH2, CH, right? And in between you have CONH2, right? Similarly, here is the chain where you have CH, CONH2, CH2, NH, CO. So CH2, CH, CH2, CH, CH2, CH. So when I say acrylamide, of course, amide is going to be there. So NH2, CONH2 is that amide component of it, right? So when I'm going to talk about acrylamide, I have CH2, CH, CO, NH2. And when I say bisacrylamide, two molecules are linked together through methylene group. So this is one molecule of acrylamide, and this is another molecule of acrylamide, and this is the methylene link, right? Are you able to see this? Right? So CH2, CH, CONH, this is one. CH2, CH, CONH, it is second one, and it is linked through CH2, the methylene. So this is bisacrylamide. All right. So when I say cross-linking of this material, acrylamide monomer is there, so you have to polymerize it. Can you just see how is the polymerization occurring? It is described as head to tail polymerization. That is, if you start with CH2, right, it is ending with what? NH. So head, tail, tail, head. That is how we describe the molecule as. So polymerization is in head to tail fashion, and long chains are going to get formed, and occasionally bisacrylamide molecule is built into the growing chain. So you have to first introduce a site for chain extension, right? So when I'm saying this chain extension, what I mean, you have to have the longer molecules. So it is bisacrylamide, acrylamide 1, bisacrylamide 2, and then you should have a long chain of acrylamide, and then the cross linking of it, so that this is going to form the pore of the matrix that is getting generated. So this is the cross link structure. Multiple molecules are there which have got which have got uh, which have got cross-linked to each other, and the pore is formed, right? Now, when I have to talk about this polymerization or cross-linking, I have to know how it is going to get formed, right? So to initiate cross-linking, you have to add ammonium persulfate. Ammonium persulfate. AP is the short form that is used, and the base NNN tetramethylene diamine, that is called as timid, right? So I'm going to the next slide where you can get to see these names, right? So acrylamide percentage, acrylamide bisacrylamide percentage, then you have the tris, then you have timid, then ammonium persulfate. These are the names, right? So this is what I'm talking about. When I say timid, it is the short form for NNNN, 4 times N, tetra, right? Tetramethylene diamine. So what is this timid going to do? We have to form the polymer. So the timid is going to act as a catalyst. So what it is going to do? It catalyzes decomposition of persulfate molecule, right? So in the second row from below, you are seeing the molecule, that is persulfate molecule. Now why the persulfate molecule is to be decomposed? As persulfate molecule, that is S2O8 persulfate, is decomposed, it is going to generate SO4 2 minus, right? So nothing but the free radical is formed. So molecule with an unpaired electron is formed. So timid will act on persulfate and convert it into a radical species. Now in this case, the free radical is going to be helpful in polymerization. How? When a free radical interacts with the matrix material M, it is say matrix material M for acrylamide monomer molecule. So this radical which is formed, so timid has acted upon persulfate, radical is formed. This radical will interact with acrylamide, right? Acrylamide monomer. Now because it is radical, you know radical is going to uh, have the unpaired electron, right? So free radical containing species are highly reactive species, right? Because they have unpaired electrons and they want to, or they need to pair with another electron to stabilize it. So the radical which is generated will react with the monomer of acrylamide and will form a single bond by sharing the unpaired electron. So this is going to be like, now if you have started with the radical saying R with the star and M stands for monomer. So R and M, they are going to interact. The R stars indicates unpaired electron. So the R and M is going to interact. Now M has shared its electron with R. So R has got stabilized, but M has become a radical species. Right. So this is again going to be RM species, but again it is with unpaired electron. So it is going to interact with another monomer of acrylamide, and then you are going to have RMM. But the second M is going to be again a radical species. That is how you are going to have formation of acrylamide polymer, right? So cross-linking by introduction of bisacrylamide molecules in the growing chain can happen, and of course oxygen removes free radicals, and we are working under aerobic conditions, right? So oxygen is going to remove the free radicals. So generally when you are going to prepare this gel, the gel must be degassed. There should not be any oxygen remaining. So the solutions are briefly placed under vacuum and you are going to remove all the loosely dissolved oxygen prior to preparation of the gel, right? So here you are going to have timid, which is a catalyst. Another way of preparing it is with the help of riboflavin, right? So photopolymerization is an alternative method to polymerize acrylamide gel. So ammonium persulfate and timid is one system, right? So it is replaced by riboflavin. When the gel is poured, it is placed in front of bright light for 2 to 3 hours. So why bright light? The light will cause photo decomposition of riboflavin and generate the free radical. Ultimately, what do you want? Free radical. So that will attack the monomer of acrylamide and the reactive species will interact with it. It will get stabilized and reactive species of M will get formed. It will attract another M, another monomer, and that is how you are going to get polymer chain formed. So either you use persulfate timid system or you use riboflavin and light. So decomposition, of course, photo decomposition of riboflavin will generate free radical, and that free radical will do what? It will initiate polymerization, right? So when we talk about acrylamide gels, they are generally talked or defined in terms of total percentage of acrylamide present. Right here in the table you are seeing acrylamide percentage 6%, 8%, 10%, 12%, 15%, etc. So the gel is talked in terms of total percentage of acrylamide present because it is going to decide the pore size in the gel. right? And by changing the concentration of both, that is acrylamide and bisacrylamide, you can decide the pore size of the gel. So acrylamide gel can be made with content of 3% and 30%. I mean the range. If you see here 6 to 15 I have shown, but it can be as less as 3 or it can be as high as 30% acrylamide. So uh, the smaller size, the smaller content 3% will give you big size or larger pore sizes, and higher quantity of acrylamide will generate pores with small dimensions or small size pores. Right. So uh, the acrylamide gels, when you are talking about, these are the basic things that you require. So you are going to require acrylamide, you are going to require acrylamide bisacrylamide percentage to be decided, right? Then you are going to work in a buffer system, which is generally a trees buffer, which is 0.375 molar said here, but it is going to vary in the range depending upon the properties. And of course, the pH is adjusted to 8.8 here. So 10% ammonium persulfate is another requirement, which is going to undergo decomposition due to action of timid, and then the radical generated will initiate the polymerization process. Right? So polyacrylamide gels, when we say, so you have to remember two things: timid plus persulfate, radical generation, or riboflavin and photodecomposition, radical generation. So radical will interact with acrylamide monomer, and acrylamide monomer will be activated, and it will act as a radical and attract another monomer to satisfy its electron uh, um, in the outer shell, to balance or to have a paired electron, right? So this is how the polymerization would be facilitated. So when we talk about proteins, they are separated on polyacrylamide jets, right? So polyacrylamide can be prepared in glass tubes, right? So generally the diameters of these gels are small, and to see the assembly you can see it in this slide. Right here you can see the vertical assembly, and then how to cast the gel. As you can see, here is a syringe, and the mixture, that is all these things that we are talking about, polymerization material. So you have to allow that polymerization to occur, but gelling should not be done. So before the gel sets, you have to uh, 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 prepare the or it gets blocked, you have to prepare the gel bed, right? So here is the stacking system, which is vertical system, and with the help of a, a syringe or needle, you can go on filling the empty space between the two slides. Right, there are two parts of the assembly, and you have to screw them, and you have to go on filling up the column, right? So uh, the vertical slabs, when you talk about that of the gels, so you can have for single run, say about 20 samples can be run together, but this is going to be like technical part depending upon the dimensions of the gel that you have prepared and the machine, the electrophoresis assembly that you have. Right. So when I talk about proteins, they are separated by polyacrylamide gel electrophoresis. So they can be separated as in presence of a denaturing agent or without uh, presence of a denaturing agent, right? So when there is a denaturing agent used, it is denaturing gel, and there is no denaturing condition, then opposite of that, that is native gel. So these are the two things. You must have heard about SDS page, right? So sodium dodecyl sulfate is the detergent which is going to denature your protein molecule, and with respect to the charge present on the protein, it is going to like just cast a shield. So entire protein will have its own positive or negative charge, but when treated with SDS, it is going to be all negatively charged all over, right? And then you are going to analyze it so that all the proteins they are carrying negative charge will get separated on the basis of their molecular sizes and the mobility as a consequence of that, right? So just to share, here I am sharing two pictures with you. What are they? I'll come to it. uh, can you just observe these photographs closely? right? This is native gel and this is SDS based gel, right? Can you see the differences? Of course, the first one is the ladder. So it is the standard molecular size, known proteins are there, and these are the samples, right? Similarly, here also you have something to see. And of course, these are the treatments given to that protein, which you need not concentrate on right now. But can you see the differences? All right. Now, here I want to talk to you about a paper just to catch your interest, which talks about use of electrophoresis. Can you imagine where you can use electrophoresis gel? And for what purpose? What kind of experiments can be done with this? Okay, separation of proteins, understood. But what more? Wherever you can find its applications, right? These are the questions. So when I say this, can you imagine the uh, 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 Antarctic fish and the normal water fish? What are the differences? You can guess what are the differences. Yes, can you tell me what would be the difference? Of course, there are proteins which are going to be shielding or important in shielding the fish under extreme cold conditions, right? So, can we take help of the methods, the instruments, wherein uh, the separation of the proteins based on uh, based on their structure, right? Based on their mm, uh, conformation, right? Its interaction with the ligands and so many things, right? They can be studied for finding out the similarities and differences in the protein. So here probably I'm trying to get that paper which I had downloaded, but I'm sorry that I'm not able to display it right now. Forget. In next lecture, I'll try to share it with you. Let's go back to the PPT again, which I actually wanted to show that a research paper, what work they had done. But in CRUSC, it is about studying the proteins uh, which are present in Antarctic fish and how these proteins are different from the proteins present in warm region or temperate region water systems. All right. So forget the application is just to compare that, but point here is when you are separating with the help of native gel and when you are separating it with the help of SDS page. So SDS page is comparatively superior. All right. That is what can be easily concluded. Right. Now, when we talk about this SDS page, sodium dodecyl sulfate. When I say this, sodium dodecyl sulfate, the electrophoresis wherein SDS page, of course, this you have learned in TY. So, nothing very great new things, but yes, summary. So, SDS dodecyl sulfate is the most commonly used detergent, and widely used method is SDS page for analyzing protein mixtures, and the analysis is qualitative analysis, right? So when you are talking about monitoring protein purification from a given source, right? So you have to separate the proteins according to their size, and the method of choice to determine the relative molecular mass of the proteins and separating them is SDS page. When I say SDS, sodium dodecyl sulfate, it is again anionic detergent when I say. So anion sodium Na plus and the rest of the chain is CH3, CH2 10 times, CH2O SO3 Na. Of course, it is really difficult to remember when I am saying, but I will repeat and you just jot down CH3 dash in the bracket CH2 bracket complete at the base of the bracket 10 dash CH2 O SO3 minus Na plus. Alright. So total how many carbons? 12 carbons. Alright. So SDS is an anionic detergent and sample to be run on SDS page. You have to first boil it for 5 minutes in a sample buffer containing mercaptoethanol and SDS. Right? What is mercaptoethanol going to do? Mercaptoethanol reduces any disulfide bridges if they are present in the protein that helps holding together the protein in its tertiary structure. Disulfide bridges, right? They need to be removed. So mercaptoethanol will do that. It will break or remove all the disulfide bridges, and SDS will bind strongly to the molecule, will denature the protein, and each protein in the mixture now in its denatured form will open up into a rod shaped structure. So you can imagine a tertiary structure protein, which is its functional form, and then you have treated it with mercaptoethanol so that all the disulfide bridges are open. So tertiary structure has gone, and then you are treating with SDS. Now the entire molecule has unfolded. Only it is like a loop or a rod-shaped structure with a negative charge on its surface. All right. So uh, s- all the molecules are going to be negatively charged, all entire along the length of the polypeptide chain, minus minus minus. Otherwise, what amino acid is there, that amino acid will have its own charge, and the net charge of the protein will depend on that. But when you treat it with SDS, entire polypeptide chain is going to have negative charge. So on an average, when we talk about one SDS molecule, it will bind to two amino acid residues, right? So original native charge on the molecule is going to be completely swamped off, will be completely removed. So entire molecule is going to carry the negative charge, right? And the rod-like structure going to remain. So it may, of course, tend to fold up, but if tends to fold up, that minus minus charge will repel each other. So you don't have the protein folding, right? Rather, the protein will remain in a rod-shaped structure. Fine. So when you talk about SDS page, two components, mercaptoethanol and SDS. Mercaptoethanol to break or remove the disulfide bridges, and SDS is to convert the native structure of the protein into rod-shaped protein. So you are going to have a sample buffer which is going to contain tracking dye, right? So generally bromophenol blue is used here also. In the slide also you are seeing the blue color. The blue color is on account of the tracking dye, which is bromophenol blue, and it will allow the electrophoretic run to be monitored. Okay, to what distance the entire sample has run. Conductivity you cannot see. You cannot see the sample that is running, but you can very well see the moving front. Then front is marked with the tracking dye, that is bromophenol blue, right? Of course, again, in the sample you have to add sucrose or glycerol, and that will change the density of the solution, you can say, sample, so that it settles in the electrophoretic buffer in the bottom of it, and then you are going to inject it into the well, so it will settle in the bottom of the well also properly. So once the samples are loaded, current is passed to the gel, and the samples to be separated are not in fact loaded directly into the main separating gel. That is what is the last point to talk about. If you see these particular gel uh, carefully, you will see one is stacking gel and another is resolving gel. So resolving is where separation is going to occur, and stacking gel is present before the resolving gel. It is very important component. When I say about the separating gel uh, or main resolving gel, prior to that you have the um, stacking gel, right? So when the main separating gel, which is normally about 10 centimeter long, is poured, so you have casted that between the two glass slides, right, or glass plates, you can say, that are the part of the assembly. Once that gel is set, then a very short gel, see, comparatively it is 1/10 of the size of the total dissolving gel size. So it is, if it is 10 centimeter, it is hardly 1 to 1.5 centimeter size stacking gel is there. So is poured on the top of the separating gel, and then it is allowed to set. Now the purpose of this stacking gel is what? Why are we making it? It is just to concentrate the protein sample into a sharp band before it starts separating, right? A system is entirely a vertical system. You are loading the sample in the particular whales, but at that time it should get concentrated, it should get what you can say, compacted, and then it has to run down the resolving gel so that you will get sharp bands, and those sharp bands then will enter the dissolving gel. So of course you have to have separating gel, and again you have to, what is the difference between these, you have to know what is the difference between the resolving gel and the stacking gel. The differences are in the ionic strength and pH. Right. So the electrophoresis buffer and stacking gel, when you are talking about, the difference is in what? The ionic strength and the pH. And very importantly, the stacking gel is with low concentration of acrylamide. So pore size is little bigger. Generally 4% acrylamide is used to prepare the stacking gel. So which is going to allow free movement of the proteins, and they will settle and sink and concentrate, or you can say this will stack. So of course, how are they moving? Not only the gravitational force, but it is under applied electric field. They are going to move, and they are going to form a sharp band. Right. So once this star band is ready, then you are going to run or allow its run in the separating gel. The band sharpening effect will of course relies on the negatively charged glycinate ions in the electrophoresis buffer. So glycinate ions are there in the buffer, which are going to help in the sharpening of the band. So they have lower electrophoretic mobility than protein SDS complexes. So lower mobility than chloride ions also. So loading buffer and the stacking gel, they are going to be important in concentrating the sample in the stacking gel. So when the current is switched on, all the ionic species will migrate at the same speed. Otherwise, you can see that if there would be a break in the electric circuit if they don't move continuously. So how to achieve this? So glycinate ions can move at the same speed, right? And as chloride ions, only if they are in a region of higher field strength they will move. So overall the buffer composition is going to contain chloride ions and glycinate ions, which are going to be important in movement of these proteins under applied electric field. So I talked about stacking gel. I talked about the buffer in the stacking gel. I talked about the components in that buffer, that is chloride and glycinate. And what is the purpose? The purpose is the ionic species will migrate under applied electric field. When the glycinate ions are there, they can move at the same speed as chloride ions only if the region is of higher field strength. So field strength is very important. And what is the relationship of field strength? Field strength is inversely proportional to conductivity, and it is proportional to concentration. I repeat, field strength is inversely proportional to conductivity, and field strength is proportional to concentration. Thus, you have to adjust the concentration of three species. What are those three species of your interest? It is chloride ion, it is glycinate ion, and it is protein SDS. So, you have to adjust their concentration. So chloride ion is going to move fastest, then protein SDS, and then glycinate. So there is small quantity of protein SDS complex. So they can concentrate in a very tight band between glycine and chloride ions. So chlorides moving fast, protein SDS moving little slow, and glycinate moving even slower. So one side you have chloride ion, another side you have glycinate, in between you have your sample SDS protein. So it will get like a sandwich ready, and concentrated form of the protein that will get concentrated in the well, and then it will move. So once the glycinate reaches the separating gel, it becomes like more fully ionized in higher pH environment, and its mobility will increase, right? So the interface between glycinate and chloride leaves behind the protein SDS complex, which are left to electrophorese. And this electrophoresis will happen at its own rate. So glycinate, once it is into the running buffer, it will start moving faster. Chloride will be moving slow then. So chloride and glycinate are almost away from SDS protein now. And the SDN protein components will start moving according to their own rates, according to their own mobility. right? So this is the overview of SDS page. Generally, 15% polyacrylamide gel is commonly used, but again, it is the molecular mass of the protein that is important, which is going to decide the percentage of polyacrylamide to be used, as you remember here in this slide we had talked about.